do that for the introduction. Like he said, my name is Javier Sánchez Utgés, and I will tell you about the work I have been doing in the Barton Group at the University of Dundee for my PhD on computational methods for the characterization and evaluation of protein-ligand binding sites. First, I will give you a bit of background, and then I will talk about 3 projects that I have worked on. The development of Fraxis, a pipeline for the analysis of fragment screening binding sites, followed by the extension of this pipeline to a larger data set and a web resource to explore it, Ligases, and finally, a benchmark of ligand site prediction, which relies on Ligases as a reference. Small molecule ligands play a critical role in protein function, acting as cofactors, substrates, inhibitors, and drugs in therapy. Therefore, identifying and characterizing the binding sites is key to understand and modulate protein function. Over the last decades, there has been an explosion in the amount of biological data publicly available. There are 5.1 billion DNA sequences in the ENA and 250 million protein sequences in the UniProt KB, most of which now have structural coverage thanks to resources like AlphaFold-DB. However, the proportion of proteins with experimentally annotated functional residues or co-crystallized ligands remains very small, accounting for 0.02% of the proteins in the UniProt KB. This underscores a strong need for computational methods to predict functionally relevant residues as well as potential ligand binding sites, which brings us to the first project, classification of likely functional class for ligand binding sites identified from fragment screening. Fragment screening is a widely used technique to identify hit molecules in drug discovery. It typically uses X-ray crystallography to determine the structure of a target protein binding to multiple small molecules or fragments. Many of these fragments tend to bind to the known functional or active site of the protein. However, they also bind to many other locations on the protein surface of unknown functional significance. These regions could be functionally irrelevant or prove to be allosteric or other type of functional sites worthy of experimental investigation. The aims of this project were therefore to define sites from fragment screening experiments, to characterize them, and to rank them based on the likelihood of function. To do this, we worked with 39 fragment screening experiments downloaded from the PDB and accounting for 1,500 structures and 1,000 molecules. 3 features were employed to characterize and find sites: evolutionary divergence from alignments, human missense variation from NOMAD, and relative solvent accessibility. In this way, 293 binding sites were defined across 37 proteins by grouping ligands by their protein interactions, and were then clustered into 4 groups by the relative solvent accessibility profile. These clusters differed in their accessibility, of course, size, and evolutionary divergence. But most importantly, they were different in their enrichment in known functional sites. Cluster 1 was significantly enriched in these known functional sites, and therefore a site with a cluster 1 label was much more likely to be functional than a site with a cluster 4 label. To conclude this project, Fraxis is a pipeline for the analysis of fragment screening sites. Grouping ligand sites by RSA returns 4 robust clusters, which differ in accessibility, size, divergence, and most importantly, enrichment in functional sites, and therefore these cluster labels can indeed be used to rank sites on likelihood of function. The second project is Ligases Web, a resource for the analysis of protein-ligand binding sites. The goals here were to extend the previous methodology to the entire PDB-E, giving rise to the Ligases dataset, and then to develop a web resource to explore it, Ligases Web, and also allow users to submit their complexes for analysis. This is how Ligases Web looks for the SARS-CoV-2 main protease. The structure panel in the center is showing the superposition of over 1,000 ligand molecules, and it can also display protein-ligand contacts for each one of the 500 assemblies for this protein. On the left, the binding sites panel displays the average features of each of the 23 sites defined on MPro. And on the right, the binding residues panel displays the features of the individual residues forming each site. To conclude, Ligases is a comprehensive dataset of characterized binding sites, ligand binding sites, and Ligases Web is a resource to explore this dataset and analyze further protein-ligand complexes. The last project is a comparative evaluation of methods for the prediction of protein-ligand binding sites. As you know, identifying where ligands bind is key to understand and modulate protein function. For this reason, over 50 methods have been developed in the last 30 years, with a better method being published every couple of months. The aims of this work were simple. Use the Ligasys dataset to benchmark ligand site prediction and compare Ligasys to previous train and test sets. For this, 13 open source methods were selected with an emphasis on recent machine learning-based methods, but also including established P2Rank and FPocket, as well as earlier methods. The most relevant benchmark of ligand site prediction is at the pocket level and can be summarized by looking at recall and precision. Methods rescoring original FPocket predictions, such as Prank and Deep Pocket, present the highest recall of 60%. In terms of precision, it is the newest machine learning-based methods, such as VN-EGNN, IF-Cypred, or GRASS, that present the best results with 95%. We also analyzed the datasets these methods train and test on. One of the main issues is that these are incomplete. Methods like PDB-bind rely on a one protein to one ligand basis, whereas Ligasys aggregates multiple ligands across structures. On this example, using PDB-bind, would only consider one of 13 known sites for this protein, leading to a suboptimal model training and an inaccurate assessment of its predictive capabilities. Another issue is that of prediction redundancy. Whilst methods may accurately predict the observed ligand binding site, often they might return multiple redundant predictions of it, which affects negatively and significantly prediction performance. By removing such redundant predictions and re-ranking the remaining ones, improvements in recall of up to 13% were achieved in our analysis. To conclude, Ligasys is introduced as a new benchmark for ligand site prediction. Incompletion leads to suboptimal learning when it affects trained sets, and inaccurate performance assessment when it concerns test sets. Redundancy in prediction results in an underestimation of recall and overestimation of precision. And rescoring of FPocket predictions by Prank and Deep Pocket achieves the highest recall of 60%. I would like to finish by thanking everybody that contributed to this work, particularly Jeff and Stuart, who are in the audience, and also Radoslav and David Coase, and you for your attention. I'll take any questions now.
One major issue of like ligand binding pocket prediction is how to split the train test data set. Like some people split it based on sequence similarity, some on structure similarity, and some say, oh, you actually need to look at pocket similarity, and if you cluster by pocket similarity, there's a strong lack of diversity in the database. So how did you tackle this issue?
Well, our benchmark is independent. We do not have a prediction method per se. What we did was to compare all the training data sets to Ligasys in order to know, how much are these methods affected by the overlap of their training set and our benchmark. And it wasn't much. It wasn't really high. And to be honest, the methods that had higher overlap between their training set and our benchmark set didn't perform better than the others.
What do you mean by overlap? Like exactly the same protein or
sequence? I mean that they trained on a ligand that was present in our definition of ligand binding sites and Ligasys.
Okay, got it.
Thank you.
Thank you so much, Javier. Next, we'll invite Alex Moorhead from University of Minnesota.